Hi, everybody. Thanks for coming back to those of you who came last night, and thanks for coming tonight for those of you who were not able to make it last night. My name is John Wilkins. I'm on the faculty up at the Institute, and it is a tremendous pleasure and honor to be able to introduce Nina Fedorov tonight for the second of these Ulam lectures. For those of you, well, I'm not going to go through the CV because, as you know, David did, I think, ample justice to her extensively honorable CV last night, and the highlights you've actually got on that little piece of paper that they handed out to you as you were coming in. But I would like to just sort of try to bridge very briefly. For those of you who were able to make it last night, you'll recall that Nina talked about, let's see, I feel like what I want to say that I came away with most strongly is, wow, have I really underestimated plants? and it's really she talked about how plants do all these things that are sort of analogous to things that we really imagine being specifically the purview of animals, that they, in some sense they see, in some sense they smell, in some sense they feel. And the thing is, I think, and anybody who works in something like plants will tell you, that of course, both in the scientific community and in our lives in general, we're very animal-centric, and we tend not to think about things like plants that much. And so very often it's the case that sort of our understanding of things that are less like us lags far, far behind our understanding of things that are more like us. And so, of course, the structure of a lot of last night's lecture was really, here are these things that you thought were special about you, but look, in the recent decades, we've started to understand how plants do similar things. One of the things that I think is really exciting about tonight's material is this is sort of a set of discoveries and ideas that have radically revolutionized our notions of what a gene is, of what an individuality is, of how evolution works, and this is actually an area where discoveries within the plant domain led the way. And it wasn't until later that we discovered, well, actually, these things, many of which she's going to be talking about tonight, are universal and apply also to animals. And I'm not going to go on and on because I think that Nina's itching to get up here and dig in on some science. But I do want to just say one thing, which, because I think, based on what I know of tonight's lecture and last night's lecture, I think that one thing that I suspect will be noticeably lacking from the content of the lecture tonight, I wanted to point out, which is, she's going to talk to you tonight about some things, discoveries in the past 100 and particularly the past 50 years. Like I said, they've really revolutionized the way that we think about life, all up and down. The thing that she is probably not going to adequately emphasize is the importance of the role that she herself has played in these discoveries and the extent to which these revolutions have depended on work that was done by her collaborators, and by her students. And like I say, those of you who saw the talk last night will have noted this sort of, it was sort of a very historical view, but if you were paying attention, there was an enormous amount of that work that was actually, she's directly responsible for. So I will not go on and on, but let me just introduce Nina Fedorov again.
Thank you very much, John. That was very nice. So, this is where we left off last night. So, this evening, we start with the fundamentals of plant sex. How plants reproduce is a bit different from how animals do it. Yes, there's an egg and a sperm and an embryo is formed when they fuse. That's all the stuff that's going on inside of the flower. These are the male and female reproductive organs. And for some reason, this is not advancing. There we go. To start where we left off. Okay. So I'm going to let this run through because it's just so beautiful. So on the right is a close-up of the reproductive organs of the flower. Those are the stamens. Those make the male sex cells. And this is the pistil. This is the female part of the reproductive organs. So let's look a little closer. This is a scanning electron micrograph of a stamen, which produces the pollen grains themselves. And if you look even closer, that's what it looks like. And each one of those pollen grains is a rather remarkable cell. Each pollen grain is itself a cell with its own nucleus. That's the central part. And inside of it, although you only see one, the very dense staining material is the sperm cell. So it's two, and there's actually two of them in higher plants. And so there are two cells inside of a third cell. Now, on the female side, this whole central structure is the pistil. This is the female reproductive organ. And at the top is a structure called the stigma. It's sticky, and that's where the pollen adheres when it lands. Here you see simply an anther close by and a few pollen grains on the stigma. Now, pollen arrives borne by wind or delivered by an insect. The showiness, the wonderful fragrances and the nectars of flowers are how they seduce birds and insects to do the work of spreading their genes far and wide. Now here's a diagram that shows the structure of the pistil itself. Here are the stigma at the top. That's where that is connected by a very long tube, the style, that collects, this is the stigma that collects the pollen, here's the style, and at the bottom is the ovary, which contains the egg cell. Now when a pollen grain falls on the stigma, it germinates. That is, it begins to send out a very long tube, and that tube gets longer and longer because the tube has to grow all the way down through this long style to reach the ovary and fertilize the egg cell. Now here's a tube, pollen tube elongating, and you see it has a little nucleus of its own that kind of supports it, but there are two sperm nuclei up here. They're called nuclei, but actually they're complete cells. And they will follow down that tube. And here actually is a picture. This is an incredible cell. It's probably one of the fastest growing cells on the face of the earth. And here you see, this is actually a time-lapse picture, but not, the frames are taken pretty close together. In fact, if you think about it, the style on a corn plant is actually the silk, and it can be a foot long, it can be even longer than that. And this pollen tube grows so fast down that tube when it lands on the silk that it can cover 12 inches in just a few hours. So on the left in the next diagram is a mature ovary. It consists of an egg cell and a number of other cells. The egg cell is right in the center. Maybe I should use this. There's the egg cell. There's some additional cells here. And this is the female reproductive structure. All of these cells are actually, have only one copy of each gene. This is what's called the female haploid generation. Plants actually alternate generations, a diploid and a haploid phase. Diploid simply means it has chromosomes from both parents. Haploid means it has only one copy of each chromosome. We'll get to chromosomes. I sometimes have to get ahead of where I am a bit. But you'll see, and perhaps you'll remember. Now, it's important for the subsequent discussion of genetics that you know that both the sperm cell and the egg cell contain only half as much genetic information as all the rest of the cells. You know that you get genes from your father and your mother. You, in turn, donate one of those genes to your offspring, which receive the rest from the other parent. The same is true in plants, so the sperm cells, as I just said, have one copy of each chromosome, and the egg cells have one of each kind. Now, when the pollen tube lands and reaches the ovary, it's growing down through the style, it delivers two sperm cells into it. One of them fuses with the egg cell, and the other one fuses with what's called a polar nucleus. It's actually a pair of nuclei. So the mother contributes two sets of genetic information, and the resulting cell develops into the nutritive part of the seed. Oops. Sorry. There we go. The vast majority of the seed is this endosperm that contains the starch and the proteins that will nourish the little growing plant as it emerges underground before it can reach the light. It must have light to make its own food, so it goes on these stored reserves. And then here is the embryo. You've probably had the experience of eating sweet corn. There's a little slippery shield-shaped thing in each kernel of sweet corn, and that's the embryo. That's the baby plant. Now, the reason that I'm telling you this is it turns out very important, a very important property which makes plants, and particularly corn. I will use the term corn and possibly maize. I use them interchangeably. Corn is known as maize in many parts of the world, and in the scientific community, it is known as maize. But the important part here is that the genetic material that's in the endosperm is exactly the same as that in the embryo. That means that the kernel is a little window on the next generation, and that makes it a marvelously useful organism for genetic studies. Now, modern day genetics started in an Augustinian monastery in the late 1800s. One of the monks, a man by the name of Gregor Mendel, did some experiments with garden peas. Mendel was a very highly educated man. He became a monk in 1843 and was ordained as a priest several days later. Then he went off to Vienna to study physics and natural sciences for a couple of years. After he came back to the cloister, he taught, and in 1856 he began the studies on plant hybridization that have become famous as the origins of contemporary genetics. He published his results in a relatively obscure journal in 1865. Now, even though hybridization was an active area of research at the time, his results got very little attention for the next several decades because they just didn't fit very well with other scientists' ideas of how heredity works. What his studies were on was the inheritance of very simple traits, such as the color of peas, yellow or green, or whether they were wrinkled or round. The reason his work didn't have much impact at the time was that the field of inheritance was dominated by biometricians. Now, this is the first meaning of biometrics. Today, biometrics means something very different. These folks generally thought that the properties of parents were mixed together, kind of like mixing red and white paint to give pink paint, so that the offspring were some kind of a blend of their parents' traits. But this isn't at all what happens. In fact, the first thing that Mendel documented rigorously was that one of the traits just plain disappears in the hybrid. I'm going to show you what Mendel saw, but I'm going to use corn simply because the ear provides such a beautiful genetic display. I will remind you that because of the double fertilization event, the kernel is genetically the same as the embryonic plant inside of it. So what Mendel saw when he crossed plants with contrasting traits like this is that one of the traits, as I said, simply disappeared in the first daughter generation, and that's called the F1. That property of disappearance of one trait is referred to as dominance. The trait that doesn't disappear, that dominates, that masks the other trait is called dominant, the other one is called recessive. Now, the second thing that Mendel documented was that the trait that disappeared reappeared in the next generation in a very regular way. 3/4 of the kernels are red and 1/4 are white. That's called segregation. Here's how it looks on an ear. Now, for the moment, you have to ignore the spots on the kernels. I'll get to those later. But you can see that basically the red and white are about 3-to-1. Now, here's how it happens. Each parent has 2 copies of the gene for the pigment, the color genes, 2 reds, 2 whites. And of course, it donates only one of those copies to the offspring. So the first generation has 1 red gene and 1 white gene, so all of them are red. Now, when you cross 2 such plants to each other, remember, only 1 copy goes to the offspring, so you can get a red gene from Papa and a red gene from Mama. That's going to be a red offspring. You can get a white gene from Papa and a red gene from Mama, and vice versa. So there's 2 ways of getting a copy of a red gene and a white gene. And then the only way that you can possibly get white offspring is by having 2 copies of the white gene. That's the segregation. Now, he further looked at the relationship of different traits to each other. We call them genes now, genetic traits. And the 3-to-1 segregation is observed for all kinds of traits. Now, here you have round and starchy kernels. The round kernels are starchy, and the wrinkled ones, that's sweet corn that can't convert the sugar to starch. The round trait is dominant to the wrinkled trait and segregates 3-to-1 in the F2 generation, as you see in the central ear. Now you can get a combination of red and wrinkled. But if you bring together both of these traits, round and red, they're both dominant, so the F1 is round and red. And then in the F2, that's the second generation, they segregate as follows. Each one segregates individually at 3-to-1. But that, if you count both traits, that gives you 9 round red to 3 white and round to 3 wrinkled and red to just 1 wrinkled white seed. So each trait in itself segregates 3-to-1, and the combination gives you the 9 to 3 to 3-to-1 ratio. Now, one of the reasons that Mendel's work didn't have much of an impact at the time is that it just struck scientists that it was just too simple. Several decades later, however, 2 very important geneticists, Hugo de Vries and Karl Correns, simultaneously did the same experiments, originally not knowing about Mendel's work. And then they started reading the literature, and despite the fact that they didn't have Google and Medline, both found Mendel's original 1865 publication and realized that they'd made exactly the same discoveries. Now, both brought attention to Mendel's work, so that by the early 20th century, the Mendelian rules had become central to the new breed of scientists. And only later were these called genes. They were called units of inheritance at the time. And only later were the scientists to become geneticists. Now, genetics became a very active field in the early 20th century, and it soon became clear that Mendel's segregation law didn't always apply because genes travel in packs stuck to each other. Corn plants and fruit flies were favorite organisms for genetic investigations, and studies early in the century established that the units of heredity, the genes, were linked together in what were originally called linkage groups. By the 1930s, it was clear that the tangled strands inside of cell nuclei, these were originally named chromosomes because they readily took up a certain kind of dye, and these tangled threads were themselves the chromosomes, and the linkage groups. Now, it isn't the case that all the genes on a chromosome always stay together because the chromosome trade pieces in a process called recombination during the development of sex cells. I'll show you that in a moment. But the essential point here is that each chromosome has lots and lots of genes on it. And they tend to be inherited together to some extent. Now, during the 1930s and 40s, when geneticists were busy mapping genes, that is, figuring out where they were relative to each other along the chromosomes, biochemists figured out that chromosomes consist both of proteins and of a long polymer called deoxyribonucleic acid, better known as DNA. And a good deal of time and energy went into arguing about whether it was the DNA or the protein that was the hereditary material. For the most part, people favored the idea that it was proteins, because they were much more complicated. They consisted of 20 different amino acids and came in many different forms, while DNA was pretty simple. It consisted of just 4 building blocks, the variable parts of which were adenine, thymine, guanine, and cytosine. You've probably seen the letters A, T, G, and C. But at just about the middle of the century, there were 2 big revolutions, the first of which was catalyzed by these 2 brash young lads, Jim Watson on the left and Francis Crick on the right. They published a little paper in 1953 that suggested a structure for DNA that made it immediately obvious how it could be the hereditary material. Specifically, what they suggested was that DNA was a double-stranded structure with its 2 sugar phosphate backbones on the outside and the bases facing each other in the center, paired up in a very specific way, such that A always paired with T and G always paired with C. This in turn suggested a very precise copying mechanism in which the 2 strands come apart and each strand acts as a template for the construction of a new molecule, and that's what you see here in this diagram. The chromosomes then are highly compacted DNA molecules wound up around proteins and further compacted in supercoils. Now, the revolutionary idea that Watson and Crick were proposing took root almost instantly because it explained so much, and within 10 years, Watson and Crick had a Nobel Prize. Now, the second revolution, which I'll get to in a minute, has taken most of the rest of the century to turn around our thinking and to turn it upside down. But let's look at the Watson and Crick revolution at a distance of a half century. Crick is no longer with us, and Watson is an elder statesman of science. This is a photo of Watson from Mariana Cook's wonderful new book titled Faces of Science. Within a couple of decades, biochemists, now calling themselves molecular biologists, had figured out the relationship between DNA and proteins. By this time, the relationship is so well established that it's called the central dogma of molecular biology. It simply says that the DNA codes for RNA, which is a copy of one of the DNA strands, and that the information in the RNA is translated into protein. Now, here's a diagram that depicts the process in a bit of greater detail. The blue double helix in the center, in the nucleus, is the DNA. And there it is being copied into the red RNA molecule, each of which contains the information for a protein. The RNA is shipped out of the nucleus to be decoded by ribosomes, these big machines down here. And here's the growing protein molecule. There are many components of it, 20 different amino acids. And the information in the RNA is, we now know, read 3 bases at a time, gradually extending the protein 1 amino acid at a time. So what's a gene? Until rather recently, it could simply be represented as a segment of the DNA that codes for a protein. The protein coding sequence is preceded by a sequence that serves as a switch called a promoter. That wasn't known at mid-century. That took another 20 or 30 years. And the promoter basically said, start transcribing the DNA here. Chromosomes, then, were thought to be long strings of genes, very much compacted with proteins, as I showed you on the last slide. This wonderful banded structure at the top right is a chromosome from a fruit fly. It's really a fruit fly salivary gland. These wonderful chromosomes develop this way because they never separate. The chromosomes keep dividing, but they stay together, and they form these big chains. And essentially, the deeply staining parts are the sequences that code for proteins, and then there are more extended connectors. Now, the representation of a chromosome on the left is an artist's idea. Of course, there isn't just one little strand of DNA down the center. The DNA there's yards and yards of it, and it's wound up very tightly in this structure, which is actually an already replicated chromosome. This is more the shape of a human chromosome or a chromosome in any dividing cell. The long extended structures get compacted, they get wound up even more tightly, they divide, and this is the last point of connection. When those points separate, the two daughter chromosomes will go to separate cells. By mid-century, the prevalent notion of evolution centered on the well-known process of mutation, recombination, and segregation. The idea is that genes change by small mutations. Keep your eye on the red and the blue genes. That's a mutation. Here's another mutation. The new, slightly different versions are shuffled by recombination. I told you a bit earlier that chromosomes shuffle parts, exchange parts. And then there's recombined chromosomes, and then they are segregated to the progeny. Other kinds of genetic changes were actually known at this time, but generally they weren't thought to contribute much to evolution. But wait, just a few years before the landmark Watson and Crick DNA paper, geneticist Barbara McClintock announced her discovery that certain genes could move to new positions. She called it transposition. But unlike Watson and Crick's DNA structure, McClintock's discovery wasn't exactly hailed immediately as one of the great discoveries of the century. To geneticists who'd made whole careers of mapping genes on chromosomes, this is kind of like saying that the kitchen could occasionally jump into the attic when you weren't looking. It just didn't fit with their daily observation that neither kitchens nor genes moved around. In fact, the variability to construct a genetic map of a chromosome implies that the genes have fixed positions and they don't change from generation to generation. So let me introduce you to this unlikely revolutionary. She was born in 1902, and this picture probably dates to her days at Cornell as a budding corn geneticist. She became famous at a young age by identifying the individual corn chromosomes. Later in the 1930s, working with a graduate student named Harriet Creighton. Keep that name in mind. She provided the genetic linkage, proved that geneticist linkage groups corresponded to individual chromosomes and that the recombination involved the exchange of chromosome pieces, as I showed you on the previous slide. But when she started talking about genes that move, that transpose from one place to another, this was about the middle of her career. She pretty much lost everyone. Other than corn researchers to whom such unstable genes were familiar, geneticists just didn't know what to make of her assertion that genes could move. I didn't get to know her until much later in her career when she looked more like this. And if you've heard of her at all, it might have been because of the stamp, which came out last year. I first met her when I was invited to speak at the Cold Spring Harbor Laboratory about some very early DNA sequencing work that I was doing. I just happened to run into her in the hall. She apologized for having missed my lecture, and I thought, why would she be apologizing to me? I was just a lowly postdoc. She invited me to come down to her laboratory, and we talked for the rest of the afternoon. I was totally intrigued. She was quick, insightful, and above all, lucid. At At the time, I was a postdoc at a sister department of the same institution that she belonged to, the Carnegie Institution of Washington, except that I was in Baltimore. So I went home and took down the Carnegie yearbooks off the shelf. That's mostly where she published. And started reading. McClintock's work was purely genetic. She had done virtually all of her work before today's molecular techniques had been invented. I thought it was beautiful, and I thought it would be fascinating to actually isolate and study these jumping genes. Now, at the time, that seemed like a far-out undertaking because no plant genes had been cloned, and actually people were saying that you couldn't clone them. About this time, I was offered a position as a faculty member at the Carnegie Institution, and my first thought was, now I can do this. It took a lot of years, and it was harder than I thought it would be, but eventually it happened. Now, what I would like to do is to show you exactly how McClintock figured out that genes jump. Even now, I find it extraordinary that she understood so much from studying just the pigment and starch patterns in corn kernels and looking at their chromosomes. Now, in the early 1940s, McClintock devised a method to break chromosomes, and she studied what happens to their inheritance patterns. The short arm of chromosome 9, which is what you see here, carries several genes that affect how the kernel looks and what kind of starch it has. She used a dominant allele of a colored gene that actually inhibited the development of pigment, so that even when there was a normally dominant allele, this was super dominant to that one. And it was designated I for inhibitor. The genes that I show you in colored blocks are ones that are required for pigment production, and then there's a third gene that I'll talk about, which is required for maturing the pigment. So, when the chromosome breaks, the genes are lost in a piecemeal fashion. And so here, when the I gene, the inhibitor gene, is lost, pigmented sectors develop. And then when the next gene is lost, it uncovers the bronze gene. So the combination of C and the recessive bronze allele give the bronze-colored sectors. So, on ears covered with kernels that looked like this, she picked up a kernel that looked like this. Quite extraordinarily different. Now, there's actually another gene on this same chromosome, and that's important genetically, but it actually doesn't show on this slide because it affects the starch composition. But we'll leave it in there because it makes the point. What McClintock figured out, what these kernels have is just two kinds of patches, and that is that they have a bronze patch and a colorless patch, no purple patches. They're just rims at the interfaces between those sectors, and they're purple, and that gives you a clue that, in fact, all the genes for the color production are still there. Now, what she found was that all of these three genes consistently got lost together, and so she named the position at which the chromosome broke. The importance of the waxy locus is that she knew that the site of chromosome breakage had to be closer to the very center of the chromosome. That white circle here is the center. It's the piece that connects the two dividing chromosomes together. There's a whole long arm of the chromosome, which I haven't represented. But what she figured out was that what was happening was that the chromosome was breaking consistently at one site, and she called that site the DS, or dissociation locus. Now, how did she figure out that the DS could move? Well, on ears, now you can see the power of May's genetics. On ears that were covered with kernels like this, she picked up a kernel that looked like this. Now, the interesting thing here is that now there are patches of color back, but then there are, in addition, these interesting little twin sectors. So what's going on here? What McClintock figured out after much careful analysis, is that this chromosome was breaking. The site that was breaking the chromosome had actually moved to a new place. Now, by that time, she had also figured out that DS didn't break chromosomes by itself, but required another gene, which she called the activator gene, and we abbreviate that AC, to activate chromosome breakage at DS. AC could be located somewhere far away on the same chromosome or another chromosome, it didn't matter. When AC sent out its signal and broke the gene, or broke the chromosome, then it uncovered these colored patches. These small bronze sectors, these small twin sectors, which are purple and bronze, however, she also figured that out. I don't have a diagram of it, but what she figured out was that the two ends of that newly broken chromosome stayed together, so that when the chromosomes tried to separate, they were attached at their centers, and they tended to break. And sometimes both of the dominant alleles would go to one daughter cell, and that would leave the other daughter cell with the recessive allele, which gives you the little bronze patches. So that's the result of one cell division, and all the daughter cells that come from one side get 2 copies of the pigment gene, and all the chromosomes that go the other way only have the recessive allele. And now, if you see the stamp, you'll know what's on it. So this came out of my own files of copies of her unpublished work. And they even reproduced her handwriting. There's the little DS, and you can see some of the genes that I talked about. There were other genes that she had on here. And this was the first diagram that she drew for herself in her own notes that showed how the DS moved to a new position. As I said, the folks that designed the stamp even reproduced her handwriting. Now let me show you how this esoteric stuff relates to more things that are more familiar to you. You've all certainly seen what's now called Indian corn. It's corn that has not only pigmented kernels, but also kernels that are variegated. I don't know whether you ever look closely, but there are stripes and spots, sometimes more of them, sometimes less of them, sometimes larger, sometimes smaller. The shapes of the spots and stripes reflect how the various tissues grow. You've all seen this kind of variegation, not just on corn kernels, but the farmer's market, but in flowers, leaves, virtually any plant part, as well as in animals, from butterfly wings to zebra stripes and Dalmatian spots. Variegation signals that a gene is working in some places and not in others. In these kernels, a gene that is needed to make the pigment is on in the colored sectors and it's off in the colorless sectors. People had tried to study the genetics of variegation for a long time, back into the preceding century, and had made some progress, but most geneticists had kind of dismissed them. Some of them even called them sick genes and thought they weren't worth studying. McClintock knew about the earlier studies, but unlike most of us, she never dismissed anything that puzzled her. She also knew that a friend of hers, and a fellow cord geneticist by the name of Marcus Rhodes, had discovered that a standard, perfectly stable, colorless allele, when combined with the right genetic background, would give this kind of a pattern of variegation. And he called the gene, he at least identified that it was a single gene that did the job, and he called it dotted because it made spots appear. So she knew that, and of course, chance, as the old saw goes, favors the prepared mind. McClintock identified a recombinant chromosome that had the DS very close to the C gene, that's C for color, and she constructed a combination that had a recessive colorless mutation on the other chromosome. So when AC activated chromosome breakage, it uncovered colorless patches. Among the large number of kernels that looked like this and had small colorless patches, she found just one that showed exactly the reverse pattern. It was colorless and had colored spots, very much like the kernels I just showed you. McClintock figured out that at the instigation of AC, the DS had jumped into the C gene and now gave that colorless background. But when DS jumped again, the gene went back to being normal, giving purple sectors. These elegant genetic experiments explained the widespread phenomenon of variegation by showing that it was due to the transposition of, in this case, DS, into and then out of a gene. McClintock announced this discovery at a major scientific conference in 1951. In her later years, McClintock liked to tell the story that people thought she was crazy when she made this announcement, but by all accounts, her colleagues simply didn't know what to make of it. By that time, she had made many important genetic discoveries, had been elected as one of the first women members of the National Academy of Sciences. In fact, the story goes, and some claim that it's apocryphal, that one of her colleagues admitted bafflement, but said that if McClintock had said it was so, it must be so. Now, another 30 years would pass before AC acquired a physical reality. Neither the knowledge nor the tools existed at the time. Even when I took up the challenge of cloning them in the late 1970s, it seemed a daunting challenge because not a single plant gene had been cloned yet. But these problems were eventually solved, and I cloned the first AC element in 1982. The first publication appeared in 1983, coincidentally, but only coincidentally, the year McClintock received her unshared Nobel Prize for her work. This is an electron micrograph that was the very first visual evidence that AC is a piece of DNA stuck in a gene. This is a DNA molecule, one strand of which came from a normal gene and one from a mutant gene that had an AC in it. The two strands stick together throughout most of their length except in this loop, and that's the AC element. It's in one place of one strand and not in the other. It's in the DNA that came from the mutant with an AC in it, and that's what it is. So what did these things turn out to be? Well, you remember that I told you that gene is basically a DNA sequence that codes for a protein. AC turned out to be a gene, but a very special kind of gene that codes for a protein that is required to transpose the gene itself. It's called a transposase. The transposase cuts the DNA and initiates the transposition of the piece of DNA that it encloses inside the special recognition sequences. So here it is snipping out the piece of DNA and inserting it in a new location. So, what's a DS? It turned out to be a C with a hole in it, a deletion. It couldn't jump on its own because it didn't have an intact transposase gene, but it could jump if there was an intact element anywhere in the genome. But the one that led to her discovery turned out to be a very special kind of DS element. It was one inserted in inverted orientation in the middle of another. Don't have time to explain it, but in fact, the reason that it broke chromosomes was because it had both sequences on both ends on both strands. So it's time to leave this excursion into the history of how transposons were discovered and get back to the big picture. I gave you this simple view of a gene as a protein coding sequence and this view of chromosomes in which the genes sit like beads on a string. Does anybody remember puppet beads? Well, that was kind of the simple picture. And that began to come apart in a serious way when genes began to be cloned and analyzed using the new gene cloning and sequencing technologies that were invented in the 1970s, 80s, and 90s. So let's start with the gene itself. The first thing I have to tell you is that genes are generally in pieces, often lots of little pieces called exons, separated by stretches of DNA called introns. This was discovered by sticking the messenger RNA molecule to its complementary DNA strand from which it was copied. Surprisingly, and this is just a drawing of what's in the electron micrograph, there were long loops, and it's the DNA that was looping, as if there were sequences in the DNA that weren't represented in the RNA molecule. So what's happening here? What happens is that the DNA is copied into an RNA molecule, reading right through the introns and the exons, but even as it's being synthesized, it's being stitched together, and the introns are clipped out, just as you see here, and the exons are connected to each other. Some genes are quite simple, having just a few exons, but at the other extreme, there are very complicated genes with more than 100 exons, and everything in between. So we're almost finished making that. Some of these genes extend over incredible lengths of DNA. So there's the messenger RNA as it's actually made. Now, one of the champion genes is a Drosophila gene. I'm not sure it's the biggest one, but it's pretty impressive. It has 116 exons, only 17 of which appear in the messenger RNA. Taken 17 at a time, the 116 exons could potentially give you more than 38,000 different proteins. And the claim is that some 18,000 of these are actually made. So think about this the next time you read about the disappointingly small number of genes in human beings. We have roughly the same number of genes as a corn plant, but each of our genes codes for many different proteins. There was a time when part of the genetic dogma said that one gene only codes for one protein. That's gone. The concept of a gene is rather more complicated than it used to be as well. Next thing I have to tell you is the genes, even in pieces, don't usually sit next to each other like little beads on a string. Not only are they complicated, but they're often very far apart. So I'm just giving you higher and higher magnifications here. Lower and lower magnifications, sorry. So each gene I'm compacting and showing the distance between them. So what's in between? Now, after all I've been telling you, perhaps you won't be surprised to learn that most of the other stuff is actually transposons. Great blocks of them. There are actually two different kinds of transposons. There are the transposons, which move as a piece of DNA, and then there are things called retrotransposons. They accomplish the same thing of movement, but they don't pick up the piece of DNA. They actually copy it into an RNA, and then they copy it back into DNA and reinsert someplace else. This clearly violates the central dogma, which says that the information only flows from DNA to RNA. Flows back again. So how do chromosomes change over time? How do they evolve? Here's a published diagram that compares a tiny section of a maize chromosome to a corresponding section of a chromosome in sorghum, a close relative of corn. The gray bars connect the similar genes. What you can see is that the genes are pretty much the same. Notice first that there are little red and purple triangles everywhere. I use little red triangles to represent the McClintock type transposons. And they're everywhere. They're inside of genes, they're upstream of genes, they're downstream of genes. These big long blocks of retrotransposons are actually located in between genes. But you can see up at the top here, two genes in the sorghum genome that look very much alike. That's actually a duplication. So duplications, transpositions, and other kinds of rearrangements separate these two closely related species. So now, the most striking difference between maize and sorghum is these blocks of retrotransposons. Very big, big, big blocks. They are just abbreviated and represented by these triangles. So in fact, they're retrotransposons inserted inside of retrotransposons, inside of retrotransposons, often uninterrupted for very long stretches by genes. Now, lest you think that plants are different from animals, take a look at this little gene region in mice and people and sheep, if you have any arrogance left. It should be gone by now. The genes are the same, although not exactly, pretty close. In fact, of your 40,000 genes or so, most are similar enough to those in mice to be recognizably related, sometimes nearly identical. It's the spaces between them and inside of them that are stuffed with transposon and retrotransposons and all kinds of small duplications, and they're different in every species. Those are indicated by the various colors in the lower diagrams here. So even though the gene sequences are recognizably similar, what's between them is very different. Here's a fascinating comparison of the mouse and human chromosomes. This gives you a good feel for how chromosome pieces get shuffled over evolutionary time. Each human chromosome is represented by a slightly different color on the right side of the diagram. The location of the similar sequences in the mouse genome is shown with the same color on the left side, and you can appreciate the scrambling. So, for example, mouse chromosome 1 has sequence blocks that are similar to human chromosome 6, 2, 18, and 1. It's a very coarse map, and the scrambling actually goes down to a very fine level. Please note that this doesn't mean that the mouse has a scrambled human genome. If you could represent its closest ancestor from which both lineages diverged in evolution, you would see that there would be a different scrambled complement of the progenitor's genes in humans and in mice. So here's how our views of heredity have changed almost unimaginably over the last century. Before Mendel, the field was dominated by biometricians who generally thought that the properties of parents were mixed together, kind of like wine and water, so that the offspring were a blend of their parents' traits. And that's true for some, but not all. So what I mean by that is that sometimes there isn't a dominant and a recessive allele, but there's codominance, that in fact the F1 is different from both parental phenotypes. Now, Mendel disabused us of the notion that this is the way genes worked, although his findings didn't really come to influence genetics until well into the 20th century. By the middle of the 20th century, the idea that there were units of heredity, which were called genes, was well established. Genes were known to be co-located on chromosomes, which recombined before being distributed to progeny. So mutation, recombination, and segregation were generally viewed as the forces that drove Darwinian evolution. This view actually persisted well into the second half of the 20th century. So another half century and many, many technological revolutions later, the view of how genes and genomes are structured and evolve has changed radically. What earlier geneticists learned is still true. Genes can change by small mutation of a single nucleotide, a single letter of the DNA's four-letter alphabet. But they also change by duplications and deletions, both small and large, as well as transposition events and rearrangements. Here's how one might think about genome evolution today. So there's a transposition event, another transposition event, a little transposition into the middle of a gene, a duplication. A retrotransposon moving, and then duplicating. It's another little batch of transposable elements coming in. But then great big pieces of DNA actually move as well. And this is how the shuffling that you saw in the mouse and human genomes occurs. Remarkably different picture. How often do any of these changes happen? Well, if you're just looking at one small stretch of DNA, you might have to wait 1 million years or so to see a change. But if you ask about the whole genome, then it's likely that some differences arise every time, or just about every time, the genome is copied. The result is that any two people in the population have different enough DNA that the forensic scientist needs sample only a tiny fraction of it to figure out whether your DNA does or doesn't match that recovered from the crime scene. So genes and genomes are remarkably plastic and changing all the time. The mystery today is why it took so long to figure this out. Why are genomes full of jumping genes able to hang together reliably enough to have convinced early geneticists that genes don't move? Now, finally, there's a growing realization that genes are not like books in a library, each its own self-contained packet of information, but more like the interconnected websites of the internet. Expression of one gene influences that of many genes and is in turn influenced by expression of others. Genes don't talk to each other directly. As you know, a gene is only an information repository, but the protein encoded by a gene can influence expression of another gene quite directly if it's involved in the immediate process of activating its transcription, or it can influence other genes very indirectly through its participation in cellular processes that in turn affect expression of genes. Nonetheless, the genes are all tied together in a web of interactions. We increasingly call these gene networks. What I'm showing you here is a network of gene interactions extracted from contemporary detailed measurements of gene expression levels, microarray data. This is work I've been doing with several physicists to ask whether it's possible to extract information about such interactions from measuring the changes in expression levels of many genes, thousands of them, over time. But what the future holds is even more complicated because each of these simple lines in this diagram represents a complex series of actors, proteins and small molecules like hormones, that carry on the conversations in cells and between cells in the organism as a whole. I end with my own personal tribute to this remarkable scientist, Barbara McClintock, who understood the dynamism of genes and genomes long before most of us were ready to think about it. She even had the concept of gene networks. I have a vivid recollection of one conversation in which she asked sharply, how is it that a mutation doesn't completely disrupt an organism? These, of course, are my words at this distance in time, but what fascinated and puzzled her was how everything in organisms was so interconnected that many gene disruptions could be absorbed and accommodated. If you think about a machine designed by a human, take a car engine. There aren't all that many parts that you could eliminate at random and still have a working machine. McClintock was a remarkable intellect, and I count myself extremely fortunate to have known and worked with her. She generously shared her knowledge and her fascinating genetic materials with me as I began to imagine that I could capture and study these elusive jumping genes. And she spared no criticism either. Although she did not know the techniques of the molecular biologist, indeed she was already long retired by the time they were invented, she stayed informed, reading papers and attending scientific conferences to the end of her life. This photo dates from the early 1980s when she first came down to Baltimore to visit my laboratory. And here she is, years later, frailer, but still understanding what these bands on gels meant and helping interpret the results. In 1990, I was approached by John Ingalls, the head of the Cold Spring Harbor Press, with the idea of putting together a volume in honor of Barbara's upcoming 90th birthday. I took on this extraordinary project with David Botstein, who was himself an early discoverer of transposons in bacterial viruses. We worried all through the project how Barbara would react, and very simply, whether she would live to see its completion. She did. And we gathered, meaning those who contributed to the book, on her 90th birthday to present it. We'd managed to keep it as complete secret. This celebration took place at Jim Watson's house. Here we are gathered on his front porch. There were many people standing around, all around the porch. Let me just point out a couple of people. Watson, by the way, was then still director of the Cold Spring Harbor Laboratory, where the Carnegie Institution of Washington, which had long since closed its genetics department, continued to maintain a laboratory for McClintock until her death. Just to point out a couple of people, remember I told you, remember the name Harriet Creighton? This was McClintock's co-author on the 1931 paper that first made her famous. This is my co-editor, David Botstein, who's now at Princeton, and this is Bruce Alberts, Barbara's longtime champion and the immediate past president of the National Academy of Sciences. And I had begun to read the preface to her. At first she joked a bit, making a few self-disparaging wisecracks. And then she grew silent, fascinated. It was a hit. She couldn't quite believe it. And though she had complained about having to dress up and go out and be with people, she said it was her best birthday ever. She was gone a few months later, leaving the world to absorb in the future what she had understood a half-century earlier. That's it for tonight. Tomorrow we take on genetically modified foods, and John has agreed
To
mediate questions. and
Yeah,
answers.
so we've got, as I understand it, 15 minutes left on the tape that they're making for posterity. So that does give us time for a few questions for Nina.
There's one right here. You have to interpret them.
Yeah, well, otherwise I'll just feel silly. Doan?
Yeah, so with all the scrambling that you described, all this chaos happening and shuffling going from kids, there must be huge amounts of constraints so that when all that shuffling is done, you can still make sense out of what's going on and still have the right body parts in the right place. And so I guess my question is, how do all those constraints work and I mean, how much do we actually understand about that?
You want to make sure everybody heard the question, understands the question?
Yeah, so the question, I believe, is roughly, if the genome really is this dynamic and there's all this shuffling going on, how can there be that much movement and rearrangement and you don't screw up the things that you basically have to do to survive?
The answer is we don't know. But the fact of the matter is, it matters more what genes you have than where they are. That's probably the simplest answer, but it's not very deep.
Other?
On that question, let's just go down to a single gene. The single gene is scattered across huge swaths of the genome. And there's a protein that's sort of running along the DNA to transcribe it into RNA. How does that protein know which parts, or how does that process say? Because it has to become a protein in the end, but massive parts of the DNA get cut out, all these things delay. How does it know which parts end up in the messenger RNA and which parts don't?
So the question is, in specific reference to what Nina was showing in terms of the introns and exons and the splicing that happens in the production of the messenger RNA, how does that actually happen? How does our body do that and actually wind up with the right thing at the end of the day?
That's really a remarkable process. There's usually an exact dinucleotide. There's a donor site and a receptor site, but these are dinucleotides, and there's lots and lots of copies of those. So there's actually a more complicated, there's a whole machine that does this, okay? And it catches the first site, cuts it, and then recognizes sequences right in the intron, and then clips it, attaches it to the next site. Now, it's not a perfect process, but good enough. And so the other part of it is that so there are recognition sequences at the ends of the intron, and there are usually recognition sequences in the middle. The whole thing forms a lariat at one point and then finishes the processing event. But as I said, mistakes are made a lot of the time because it's not constrained heavily. And so there are layers of proofreading. And one of the most remarkable things that has been discovered relatively recently is that if the coding sequence for the protein isn't right, the RNA gets trashed before it gets out of the nucleus. So there's this huge turnover. Much of the RNA that's made is degraded. It's proofread repeatedly, and only if it's right and the coding sequence is intact does it get out. It is a really awesome process, and I can't say that we understand every detail of it, but it is remarkable.
Yes, yes, you.
There's been research done with biophoton emission from the DNA cell as a possibility that it communicates with the cells. It's a communication device. Have you looked at that at all? Do you have any comment on that?
I'll try to translate that
Yeah,
because
I'm not
I'm not
sure I
familiar with
understand
that,
the
so I may or may not get it right. But it's a question of biophoton emission from the DNA
Right.
as a mechanism of communication to other parts of the cell.
Yes. Or other parts of the cells in the body. Not just-
You stumped
So
me.
to other parts of the cell.
So you're
or other
saying that
cells.
it's actually light that is emitted by the DNA
Right.
that communicates to, you've stumped me. I'm not even familiar with that.
I wanted to ask if you know anything about the methylation theories that they have on how methylation is now controlling pseudogenes and transposons in the genome, especially when it comes to Shepherd's
You
protein.
got another hour?
So the question is if Mia could comment on the process of DNA methylation, which is a chemical modification of the DNA, and its role in actually controlling the movement and the reshuffling of these various transposable elements.
The quick answer is yes. That's now a whole field of epigenetics, but I will tell you that that's one of the things that McClintock actually was one of the two earliest observers of. And that was a very active area of research in my lab. And basically what she had reported, and the other person, by the way, is Mary Lyon, who studied X chromosome inactivation. Basically, what McClintock observed with transposons is sometimes they would disappear. They would go out as if they'd been deleted. But then a few generations later, in a few kernels, they'd come on again. So to make the long story pretty short, what we understand today is that there's a whole another layer of genetics, which is called epigenetics. And it's that inactivation is mediated by the production of little tiny RNA molecules. They're called silencing RNAs and microRNAs. And they go back and they trigger a methylation and an activation, so a folding up of that piece of chromosome. So part of the answer to why geneticists didn't see these transposons is that they're silenced, folded up, and recombination doesn't happen. It happens in the unfolded gene regions. So they act like they're not there, and they're very stable. Very interesting area of research. And it wasn't, in fact, when I started working on it, it was really, anybody that worked on methylation couldn't possibly be considered a serious scientist. And now it's a whole field. It's really interesting.
You mentioned in Drosophila there was a case where there were 116 somethings, of which 17 were taken out to make something. Well, how deterministic is this? How do you get the 30,000 or whatever? Do you start at different places? What's different in these two or 30,000 different combinations of 17 things?
So the question here touches back to the earlier question of the splicing of the messenger RNA and the connection of the exons to make the complete messenger RNA. And in these ones where you've actually got this sort of combinatorial process, right, where there are many, many exons and you select a subset of them and compact them together. How deterministic is that? How well regulated, and how are those 17 actually selected from those 1,000 some odd exons?
That's a really active area of research. And let me give you the simple answer. And that is that you can imagine that if you can go from exon 1 to exon 2, you might be able to skip over exon 2 to exon 4, right? And so the process basically makes alternative combinations of the exons. And how well regulated it is, there are cases, for example, the sexuality, the femaleness or maleness of Drosophila is determined by alternative splicing. Extremely well regulated. In other cases, we don't know. There may be a background noise level of incorrect processing that is then trashed. In other cases, it may be very well regulated. The thing that really it makes it very difficult to nail down, especially in humans, is that it's very hard to figure out whether a particular combination is actually functional, or whether you can detect it in the RNA. But how do you figure out when you have multiple overlapping things like that? How do you do genetics on it? The other element, I've talked to you about AC, but the element that I've studied most thoroughly of McClintock's elements is an SPM element. And we discovered that this one little gene had 4 overlapping transcripts, and we had to give up classical genetics because all we got was deletion mutations. So what we did was ended up reconstructing, taking 2 of these transcripts at a time, and asking which pair was required for transposition. Those are experiments that are very difficult to do in people, but you can do them in plants.
Yeah, are there some gene clusters that strain and very rarely or never interfere? For example, the homeo, the genes that are required for developmental changes, like the Hox genes.
The question is, are there some genes or clusters of genes that have such an important function, or maybe a function that's not robust to this type of translocation, where actually this rearrangement process or translocation process is silenced in particular regions of the genome?
That's a really interesting question. I'm not sure that was the question, but however it came out. Okay, I think that what he was asking originally was, are there genes that are so strongly concerned that they can't change? And the answer to that is yes. There's a full spectrum of them. There are some that are so recognizable, they're conserved by function. So for example, plants have hemoglobin genes and people have hemoglobin genes. What's a hemoglobin gene? It's a gene whose structure interacts very strongly with oxygen, and because it's actually mediated by an iron molecule. But the molecule is so similar, it's so constrained by the necessity to bind oxygen. Although plants and animals use them for completely different reasons, for really different purposes, but they're recognizably similar. There are other genes that are almost identical, and there are others that diverge very rapidly. So the other question, your question, or rephrasing of his question, was, are there areas of the genome where the arrangement is so important that they're left alone? The answer is yes, and moreover, it's even more interesting than that, because recently it's been discovered that there are portions of the genome that evolve faster than others. So by measures of single base changes, transposons, rearrangements, everything else, they have lower rates of change than others. How does that happen? Don't know. Very interesting.
I've often wondered, when the DNA is packaged inside the cell, it's an incredible problem that a cell has solved to be able to package it that way. And then as cells change and divide and grow and mature and differentiate and die, it would seem that as they're going through these different stages that you have to continually package and repackage. And I'm just curious if, while the transposons and the duplications and everything is going on, if there's like certain parts that sort of like get unwrapped, and then you get more mature, and then maybe something else I know that's really simplifying it, but I'm just wondering like if parts of it get unwrapped as we age, as the cell is dividing.
Well, until you got to the age part, I was just going to say yes. Indeed, the whole machinery that does this, that allows parts of it to unwrap and actually separates DNA strands, is remarkable. So there are enzymes that actually allow strands to penetrate. So they'll divide the DNA, hold on to the ends, and then seal it up again. That's happening all the time. And the remarkable thing is that if you knock out one of the enzymes that is constantly reconnecting the DNA, even as it's replicating and dividing and getting repackaged, the genome gets scrambled almost immediately. A fellow by the name of Fred Alt has done some incredible experiments where he essentially painted the chromosomes with fluorescent dyes, different ones, and then watched what happens to these chromosomes when you knock out an enzyme that reconnects blunt broken ends. And almost immediately you get scrambled chromosomes. So this is a process.
We need to stop and give Nina one more round of applause and encourage everyone.
And what a great audience.
And I would like to encourage everyone to come back tomorrow night. There's one more lecture in this series.